Hi everyone, this is Dr. Asa here, and channel Dr. Asa Explains. Today's topic is Mendelian genetics. Okay, so Mendelian genetics is the basic genetics. It starts with basic vocabulary that needs to be studied and learned before actually the Mendelian rules and theories can be understood. So some of the basic vocabularies are gene, allele, diploid, haploid, dominant, recessive, fertilization, gamete, genetic recombination, genotype, phenotype, haploid, diploid again, heterozygous, homozygous, hybrid. These are some basic vocabularies. So we will start with uh, Mendel and his laws of independent assortment and laws of segregation. So Gregor Mandel is considered father of genetics. He's actually he started some of the very first experiments and published one of the first scientific papers on the genetics that we know genetics today. So, like any geneticist today, he actually used a model plant. His model plant of choice was pea plants, garden peas. So he picked a model plant pea, but it was a good model plant because pea actually has diverse variation of flower colors, leaf, the stem length, a lot of other features, seed length, seed shape, seed size, seed colors, so that actually they were really good fit for his studies. So when he actually start just hybridization, crossing these different different varieties of the peas and look at the from parent to F1, F2, F3, these different generations. So he actually came up with these results that actually today we still use in typical genetics. So as we know, gene is the hereditary unit transmitted from one generation to next generation. So, um, and it's we know that gene actually, instead of like some early beliefs, gene Mendel did not call it gene, but he actually discovered that something moving from one generation to the next generation, and that's actually we call today gene, which has different um, versions we call allele, like eye color, or maybe blue eye color versus green eye color or brown eye color, right? So, and then these are the expressed traits of the same gene. So, alternative form of a gene um, is allele, and genes are always in pairs. So, um, now genotype is the genetic makeup of an organism. Phenotype is uh, phenotypic physical features of an organism. And the phenotype is a combination of genotype times environment, which is another factor affects phenotype, which could be nutrition, temperature, physical environment that our organism lives in. Okay, so that's the express results of a gene in a given environment, what we call phenotype. Phenotype can be wild type, which is this normal allele, most common allele, or mutant, the very rare altered allele, less common, often recessive allele. We call so that's actually we call mutant. Alleles can be dominant or recessive. So dominant always express, recessive alleles only express when the big A or dominant is missing. So generations are P, which is parental, F1, which is the first filial gene, which is P1 times P2, F2, which is the subsequent generation, which is F1 times F1, F3, and it goes on. Okay, so test cross is a test which is basically to reveal the unknown genotype. So if you have an unknown plant, let's say, if you want to find out what's the genotype of it, you just hybridize it, cross it with a um, little p, like an homozygous recessive of that phenotype, and then look at the F1 results. If if all results are purple, which we know that um, that must be the unknown must be homozygous. If half is half a half distribution, then we know it must be heterozygous. So dominant can be in three different forms. When we say just dominant, it really means most of the time completely dominant. But there is also two other forms. One is incomplete dominant, like we see in heterozygous as an intermediate blending phenotype, um, or we have codominant, like in blood types. Basically, both alleles can be observed phenotypically, like AB blood type. And there are also multiple alleles, and there is something called epistasis if more than two genes actually, more than two alleles for the same gene found in a population, and then one, so I mean more than two genes, and then one affects the expression of the other one. So and you need to know problem solving and distributions of these ratios, one gene, two genes, or several genes. We know that 3 to 1 is typical one gene distribution, 9, 3, and 3, 1 is typical two gene distribution. Multiple genes, we don't have, we could find the ratios, but what we call is we use this QTL, quantitative trait loci. So please go ahead and study the exercises in your book on Mendelian genetics, and then complete these basic exercises, and then work on the vocabulary. And then if you have any more further questions, please reach out to me. Good luck with your studies, and see you in the next video.